Hi everyone, this is Ginny LaBelle with Defined Health, and we welcome you to today's Insight Series webinar, The Dash to Treat NASH, Looking Toward the Finish Line. Today's webinar will feature two members of the Defined Health Senior Consulting Team, David Lom, Associate Principal, and Brent Osborne, Consultant, as well as the two panelists, Roger Longman of Real Endpoints and Manal Abdelmalek of Duke University. I want to let you know from the Defined Health side that we've scheduled our Cancer Progress meeting for 2018 in New York on May 8th and 9th. So make a note of that. You'll be receiving more notices about that. We hope you'll join us there. We will also be participating in a couple of upcoming events in September, CNS Diseases Summit in Boston, GTC's meeting, and Biopharma America in September, late September 26th and 27th, and we hope to have an opportunity to see you there as well. To learn more about us and our early strategic commercial development services, give us a call, send us an email, or visit our website at definedhealth.com. For today's webinar housekeeping notes, we hope to have time to field questions toward the end of the session, but please send your questions as you think of them, and we will answer them along the way or at the end of the session. If we don't get to them, we'll follow up with you directly in the next day or so. We anticipate that the slides and an audio-visual or an audio version of this webinar will be posted on the Defined Health website within the next few days. Thank you. Please enjoy the webinar, and I'm going to turn it over now to David.
Thanks very much, Ginny, and welcome everybody today to the webinar and panel discussion, which again is titled The Dash to Treat NASH. It's looking beyond the finish line now. So I'm going to be moderating the webinar and panel discussion. Again, my name is David Lom, along with my colleague, Dr. Brent Osborne. And we're very excited today to have the two panelists that Ginny mentioned very briefly there, Dr. Manal Abdelmalek, who is an associate professor of gastroenterology and hepatology, and also the director of the NAFLD clinical research program at Duke University. Manal is going to be sharing with us her perspective as a clinician on the current diagnosis and management, I should say current and future diagnosis and management of patients with NAFLD and NASH. We're also joined by Roger Longman, who is the Chief Executive Officer of Real Endpoints, as Ginny mentioned. That's a company that specializes in or focuses on pharmaceutical reimbursement. And Roger has a long history and great experience in this area. So Roger's going to be helping us to understand the payer's perspective on the management of drugs for NASH that we hope will soon be approved, which is a perspective I don't think we've yet heard enough about within the community. So just to get us all started, and I'm going to ask Roger and Manal to introduce themselves, but we'll do that once we get through some of the background material. So I just wanted to start by reviewing Defined Health and my experience in this space. So Defined Health has been really following developments in fibrosis in general and NASH in particular since sometime around 2012. And since that time, we've conducted numerous evaluations covering multiple indications associated with fibrosis, IPF, PBC, PSC, renal fibrosis, and many, many others, but quite a few evaluations specifically focused on NAFLD and NASH. In addition to that, we've done a number of webinars and workshops and panel discussions, some of which are listed on the page here. Most recently, we did the NASH workshop at the NASH Summit in Boston, along with Dr. Abdelmalek and Brent Osborne as well, my colleague. But I wanted to point out that since 2002, when we first started covering this space, things have really taken off. They've really begun to have evolved since that time. I think at our first evaluation, there's somewhere around 50 agents in development for NAFLD NASH, maybe a little bit under that. And now we're up to over 250. The deal activity certainly has, increased its pace. A lot of activity in the space over the last few years, a lot of very large deals. And over that time, the forecast of the commercial potential of this opportunity have really risen dramatically as well, from the low billions to now some analysts forecasting that the market may be worth 35 to 40 billion worldwide for NASH. So, we started raising a number of important questions early on, and some of these questions have been addressed, such as, at least partly, such as the clinical and regulatory path. We now have a better idea of what that's expected to look like. The ways in which patients may be identified in clinical practice, non-invasively, we have a better idea there. But there's a lot of questions which I think remain to be, that should be discussed more thoroughly, that perhaps haven't been given enough attention yet. One of these has to do with the size of the addressable patient population, because we believe that the addressable population may actually be much smaller than population-based studies may suggest, which of course has important implications for the commercial opportunity. And then I think the payer issue is another question which really requires some more exploration. So, will payers exert any pushback once drugs are approved for NASH, and how will that impact management of those drugs? We certainly have a number of important examples to look to, which I think Roger will speak to, the PCSK9 inhibitors for hypercholesterolemia, drugs for HCV, but certainly payer pushback may be an important thing to consider. So before we get to the discussion and before I have Roger and Manal introduce themselves, I just wanted to set the stage by talking a little bit about what exactly NAFLD, non-alcoholic fatty liver disease, is and what NASH is and why it's important. So non-alcoholic fatty liver disease really encompasses a spectrum of disease ranging from simple steatosis, just fat in the liver, which is pretty benign, to NASH or steatohepatitis, where we have inflammation and some degree of fibrosis, all the way to cirrhosis, where we have significant impairments in liver function and potentially to HCV. But what's important to realize, again, is that this is a spectrum of disease. So not all patients with NAFLD will develop NASH, and not all patients with NASH will develop a progressive disease or more advanced disease. I think that's very important to consider when we think about the patients that might be amenable to treatment. And the text that's bolded here really points out, it makes an important point, which is that fibrosis stage, but no other histologic feature of NASH, is independently associated with long-term overall mortality, liver transplantation, and liver-related events. So that's an important thing for us to think about, is the degree of fibrosis in these patients. Now, the next slide I'm just going to go through very quickly, but I did want to touch on pathophysiology, and really it's not entirely understood as of yet, but most experts believe that a two-hit or multiple-hit model best explains the pathophysiology of NASH, starting with insulin resistance, leading to steatosis, and then some second hit, such as cytokines, adipokines, etc., especially oxidative stress, may act as that second hit, which results in the progression from steatosis to inflammation and then fibrosis. And again, that's important when we think about the patients that may progress. So the next two slides really get at the point of why is NASH important? Why should we be talking about this? Well, for one thing, NASH is expected to become the most common cause of advanced liver disease in the coming decades. So prevalence of NAFLD is highest in populations with pre-existing metabolic conditions, so obesity, type 2 diabetes. We, of course, know that the prevalence of these conditions is growing, so it's expected that the prevalence and impact of NAFLD will grow as well. And today, studies estimate the prevalence of NAFLD at about 10 to 35%. That equates to about 70 million people in the United States with fatty liver. But remember, not all of these patients will develop NASH, which is the more significant part of the disease. It's only about 10 to 30% of these patients develop NASH, but that's still about 16 million. And one additional point is simply that not all of those patients with NASH will have significant fibrosis, which again is speaking to the addressable patient population. But we'll get there in a minute. The other point is that NASH is expected to become the leading indication for liver transplant in the next 10 to 20 years. So the two graphs on the slide simply show at the top its annual trends in liver transplant by diagnosis, This graph only goes up to 2009, but you can see that hepatitis C clearly is the leading reason for liver transplant, but that is starting to decline somewhat even by 2009, and we can see the bottom solid line and the dashed line atop that, which account for the patients with NASH, that's starting to increase. And if we look at the graph below that shows annual trends in new liver transplant waitlist registrations, that goes out to 2013, we can clearly see the solid line here again is NASH, and it's clearly picking up where HCV peaked around 2011 and starting to decline. So it's understood that NASH is going to be an important cause of advanced liver disease, an important cause for indication for liver transplant. But it's important to point out that most of these patients still aren't dying due to these liver-related events. They're dying due to cardiovascular disease. You have to remember these patients also have, they're obese, diabetic, have metabolic syndrome, many or most of them, that is. So cardiovascular disease is still the most important cause of death. And again, this is going to have important implications for when we think about treatments aimed specifically at NASH and the cost effectiveness of these treatments. So in order to understand the addressable patient population, I think it's important to walk through the diagnosis of these patients. So the table, or excuse me, the flow chart on the right here really just shows how in clinical practice today, most patients with NAFLD and NASH are being picked up and diagnosed. So it really starts typically at primary care. Patients are going to be identified based on abnormal liver function tests in the context of obesity, insulin resistance, standard on routine exam. They may then, if there is an issue, be referred to a hepatologist, or they may get ultrasound before that, and imaging or other imaging to screen for fatty liver. And once fatty liver is detected, you can say that the patient has non-alcoholic fatty liver disease. Next, you have to rule out alternative causes of fatty liver. So you have to see about the patient's alcohol intake, presence of HCV or HBV. And then what you'd want to do, most clinicians are doing, most hepatologists are doing, is assessing the patient for risk of significant fibrosis. And they can do that by looking at clinical features, again, the presence of diabetes, metabolic syndrome, other clinical factors such as platelet count or age. You can put this into an algorithm, or you can just sort of gather the clinical gestalt. But we also can use a non-invasive technology such as FibroScan, transient elastography, which gives a sort of an idea of the stiffness of the liver, which correlates to the degree of fibrosis. And the main point here is that today, liver biopsy is still required to make a definitive diagnosis of NASH, but it's used very infrequently, certainly less than 10% of patients, probably much less than that. The clinicians are most concerned about the liver fibrosis because that's what correlates with the poor outcomes. Now, you know, we clearly can see that NASH is an issue. It's a problem, but as of today, there are no drugs which are FDA approved specifically for the treatment of NAFLD or NASH, and there's no universally accepted pharmacologic standard of care. So I put in this flow chart here, but to be honest, most of the treatment is really lifestyle modification aimed at weight loss, physical activity, increased physical activity, and then treatment of any associated metabolic risk factors. That's really the primary treatment for most patients. There are a few drugs which can be used off-label, such as pioglitazone and vitamin E, have shown some benefits, modest benefits, but also have some safety issues, potential safety issues associated with them, so they're not used all that often. And then patients with cirrhosis, it's really about surveillance for HCC and other issues. But I just want to stop and recoup here for 1 second. So, we're talking about 16 to 30 million adults affected in the U.S. alone. No FDA-approved treatments. Based on this, a number of analysts have proclaimed NASH to be the next big global epidemic, the next hepatitis C. And now analysts are forecasting that the market for NASH treatments, as I said before, could reach 35 to 40 billion by 2025. So it's really a huge market, looks to be untapped commercial potential, and it certainly hasn't been lost on drug developers. I'm going to turn it over to Brent Osborne, who's going to walk us through the pipeline and some of the deal activity now.
Thank you, David. So while there are no FDA-approved therapies specifically for the treatment of NASH or NAFLD, there are a lot of assets in development. And as was mentioned at the beginning of the webinar, when Defined Health first began evaluating this space about 4 or 5 years ago, there were less than 50 agents in development. But now, as you can see on the slide, there's significantly more than 200 and upwards of 250 agents being developed in this space. The majority of these are still preclinical. However, there are about 82 agents in the clinic at some point right now. And this just illustrates how much interest there is in this space. And in addition, there's a variety of different mechanisms being pursued, including repurposed anti-diabetics, anti-steatotics, anti-fibrotics, FXR agonists, of course, and others. Now, one of the values of having mechanistic diversity within the development pipeline is that different stages of NAFLD, NASH pathogenesis can be therapeutically targeted. For example, some agents are able to address simple steatosis, whereas others have been reported to have effects on inflammation and scarring. Some therapies are more challenging to bucket in this way and may have more of a pan effect with the potential to improve multiple clinical parameters along the spectrum of disease here, including, interestingly, the most two advanced programs, Intercept's obeticholic acid and Genfit's elafibranor. So this should begin to highlight the importance of careful clinical evaluation of each patient and the need to tailor the right treatment or potentially combination therapy to each individual patient. And this is something I'm sure we'll address a little bit later with our panelists. So with that said, most of the drugs in clinical development are targeting patients with biopsy-confirmed NASH, including evidence of at least some fibrosis, that being at least F1. However, most of these trials seem to be structured to include predominantly F2 and F3 NASH patients. So along with the increased development activity, we've seen a concurrent rise in deal activity in recent years, with several notable M&As and partnerships that have been negotiated. Now, I won't go through all these details, and the slide just represents some selected deals, but you'll see most of this deal activity is occurring prior to phase 3. And after some release of value-inflecting data, and in typical fashion, Big Pharma is acquiring or licensing assets for a significant upfront payment and subsequent milestones as the program continues to progress. So despite the volume of deal activity, we still have a lot of unknowns regarding how large this market really may be, as David alluded to earlier, and I'll turn it back to him now to discuss this further.
Right, so this is really getting to, and thanks, Brent, to the two main topics of discussion, I think, today is, one of them is really the size of the addressable patient population. So how are clinicians going to choose the correct patients for treatment? How are they going to identify them in clinical practice? And then the other issue is the payer issue. So the first, this slide here really addresses this first point. So, the population-based estimates do suggest a very large number of individuals with NASH, but we believe the addressable patient population actually may be much smaller, at least today. So as I said at the beginning, we've got about 70 million patients with NAFLD, but the way patients with NAFLD are typically identified is by routine blood tests showing elevations in liver enzymes. It's important to note that liver enzymes are not elevated in all NAFLD patients, and many individuals don't undergo routine wellness exams. So just to start off with, many patients with NAFLD are not being diagnosed. Then it's only 10 to 30% of patients who would have NASH, and only 25 to 40% of these, and these are all rough estimates, so we don't have great data on this, that will progress develop progressive liver fibrosis. And right now, only a small percentage of patients are undergoing liver biopsy, and the non-invasive methods that are routinely used in the clinic, or that are sometimes used in the clinic, I should say, are not very good at identifying patients with very early fibrosis, so things like FibroScan. So the number of NASH patients currently being diagnosed, we believe, may actually be much smaller than the population-based estimates would suggest. And it's important to point out, though, that current low diagnosis rate, that's at least partly due to the lack of effective treatment. And certainly the diagnosis rate could increase with time as we have drugs that are more effective and approved for this disease. The second point I wanted to make then is just the impact of payer management of drugs for NASH. So it's certainly possible that payers could restrict access to NASH drugs, especially if they're priced such that they don't meet current willingness to pay thresholds. And I'll just say here, if they're too expensive, if they're perceived to be too expensive. We don't need to go through all the details in the slide, but really what this lays out is simply the draft guidance or preliminary guidance that was issued by the Clinical and Economic, the Institute for Clinical and Economic Review, ICER, which did look at obeticholic acid, Intercept's obeticholic acid, both for PBC, for which it is already launched, and for NASH, which it's currently in development. And the two tables down at the bottom there are just showing their cost-effective analysis at two different prices. The top is $65,000, which is near the price that Intercept launched OCA for PBC. And the bottom is $15,000, which is much closer to what most Wall Street analysts are predicting OCA might be priced at if it's approved for NASH. And you can see even in the table on the bottom, the ICER, the dollars per quality, are still considerably higher than the willingness to pay threshold of $100,000 in the U.S. When the price is $15,000 per year. Now, I have to point out that this analysis is based, it's a preliminary analysis for one, it's based on data from the Flint study, not the phase 3 trial, so certainly the analysis may change with more mature data. And in addition to that, ICER's recommendations, of course, are not binding in any way, but they may provide some ammunition for payers to push back on drugs such as obeticholic acid once they're approved. So now I want to turn to the questions for discussion, because I think we've established that this is a relatively large, well, it's a large market. Exactly how large the addressable patient population, I think, still remains to be determined. But there's a number of important questions that we want to discuss with the panelists today. Starting with what will a label indication for NASH look like? I think we have some sense of this, but we can address this very briefly. What's more important is we want to understand how are clinicians going to determine which NASH patients to treat. Are they going to base their decisions based on the level of fibrosis, clinical factors, other things? And then how are clinicians going to identify these patients in clinical practice? Importantly, we want to turn to Roger to focus on the criteria payers might use to determine eligibility for reimbursement. And then, of course, how will the pricing of drugs for NASH influence market access and reimbursement? And then we want to talk a bit about combination therapy. So most experts believe that ultimately combination therapy will be required. So how would payers manage multiple drugs for individual patients? And the last point I want to talk about is what can companies developing drugs for NASH do to maximize their share of the market and to facilitate access and reimbursement? So I'm going to start with the first two questions, and I want to turn this over to Manal first and ask Manal if you can give a brief introduction. We'll turn it over to Roger afterwards to give an introduction, and we can walk through the questions then. Manal?
Yeah, so the first question is, what will the label indication for NASH look like? And how will clinicians determine which patients to treat? So I think the labeling, in my opinion, is really going to look like the phase 3 registry study inclusion criteria. I don't think that the FDA will liberalize the labeling or introduction of these compounds outside the scope of which they were studied. And thus, if the study cohort was patients with biopsy-proven NASH and stage 2 or 3 fibrosis, that will be likely what the label would otherwise indicate. Now, gets into a different discussion whether once approved, use and access to such drugs will be extended beyond the label, and that will boil down to a cost economic strategy. But in my opinion, the label will be reflective of the cohort in which the compound and drug was otherwise studied. I do not anticipate that the label will liberalize our risk stratification or diagnostic approaches to determining who has access to drug. If the study is deemed biopsy-proven NASH, I doubt the FDA will allow for a surrogate marker of disease unless that surrogate is well validated against histology at the time the drugs reach Subpart H approval.
So, Manal, that's a great point that I wanted to just follow up with very quickly, because of course the registration studies for obeticholic acid, for GFT 505, we're looking at primarily patients with biopsy-proven NASH, primarily with F2 and F3 fibrosis with, I believe it's an exploratory cohort or a smaller cohort of patients with F1 fibrosis. So, what we're really interested to hear is in clinical practice then, how would you use the same metrics to determine which patients you will treat? In other words, only F2 and F3 fibrosis, and if so, how are you going to identify those patients? Would it be biopsy that you would be expecting to do?
The unfortunate reality is the only gold standard that has yet to be really used, and we can argue whether it's golden or not, is liver biopsy. And so, yes, I do anticipate, but hope not to have to utilize liver biopsy for the long term. There is a lot of work going on in biomarker discovery, both blood-based as well as imaging biomarkers, and we are getting there, but I do anticipate the use of liver biopsy. I also do anticipate that the FDA as well as clinicians will be risk stratifying patients based on the severity of their liver disease, and that based on the health economics of the compounds that are approved, if they come into market very, very expensive, then it certainly will be the highest need patients, the highest risk patients, those with advanced fibrosis that would otherwise be given liberties to access to these compounds. However, if compounds come in and they're very safe, very inexpensive, then I could anticipate that irrespective of risk stratification, that such compounds could be used earlier in the disease state, particularly if they have positive influence on other clinical endpoints, such as how the patient feels, how they function, how they survive from all-cause mortality, and particularly even cardiovascular-related morbidity and mortality. Because the presence of NAFLD and NASH not only increases cardiovascular risk, but also is a predictor of new-onset diabetes. So how broadly these compounds are going to be used is going to ultimately boil down to cost, safety, and not only liver-related efficacy, but overall all-cause morbidity, mortality metrics.
Okay. And Roger, I'm curious if you have any comments, and maybe you can give an introduction before you join in.
Sure. I'm Roger Longman. I'm the CEO of a company called Real Endpoints, and we focus our attention on market access and reimbursement. We have a number of proprietary tools that assess the value and the economic impact of drugs on payers and health systems and patients. And we use these in our to figure out development strategies, launch strategies to maximize the value of drugs.
Sure. Well, responding to some of the comments, if you could respond to some of the comments that Manal made. So we're talking about first of all, who she would treat as a clinician, and the idea that she would probably treat according to, close to the registrational trial guidelines or the enrollment criteria, and that this would be F2 and F3 fibrosis. We've also got, again, an exploratory cohort of patients with F1 there. But these are liver biopsy-proven NASH patients. Now, I'm curious from your perspective, from the payer's perspective, what sort of management of a drug, and let's say we're talking about something like obeticholic acid, so it's a drug, or GFT 505 for that matter, drugs that have, modest efficacy They're fairly safe, may have, some minor issues, obeticholic acid, of course, with the pruritus and the elevations of LDL. How do you, how do you think payers would react to a drug like that if priced at, somewhere around $15,000 per year, as many analysts are suggesting? How does that get managed then?
Well, that's a big question, and there are a number of aspects to it. But let me just start with Manal's point about the label. The first thing I think you have to understand is what labels mean to payers.
Sure.
And it's not the same thing that labels mean to physicians or medical groups or pharmaceutical companies. A label is a starting point for a payer, and it's a document. It's more or less a literary document in a funny way, because basically or philosophical document, because basically what many payers do is interpret labels, and interpret labels to their best interests, and generally those interests are economic. If the label really turns out to be, as Manal was saying, a biopsy-proven NASH in people with, F2 or F3, I think payers are going to be fairly strict about insisting on that. And when I talk about being fairly strict, you should think about, what's happened in the past. So if you think about the PCSK9 labels, for example, as I recall, it was something like, heterozygous FH or ASCVD in patients who are on maximum tolerated statins and require additional LDL-C lowering, something like that. And so what plans did was they said, well, what does additional lowering requirements mean? And so you saw some plans say, well, additional lowering means anybody that you have to have an LDL-C greater than 130, which is way beyond what most people would have said. And then when they said, well, what's the definition of a maximum tolerated statin? Well, okay, let's demonstrate that by saying, if you don't have rhabdomyolysis or elevated CK creatinine myoglobin or something like that, you're not going to be able to get this drug. And when they say you have to be on statins, they said, well, all right, you have to document adherence to statins, and you have to document that for a number of months before prescription. And so the notion that a payer looks at a label and says, all right, I'll just go along with that label, sort of belies the fact that what they really do is they interpret that label, and they interpret it from their point of view. The second thing that they're going to do is they are going to follow where they are pushed, or they're going to move in directions to where they're pushed. And they can be pushed in, by a variety of different forces. But if there is, but in some, if there is not a significant cry from the most influential groups, the most influential medical groups, or for that matter, from patient groups, they're likely to be fairly restrictive at reimbursing these drugs, unless there is some overwhelming economic argument for doing so. And that's not clear to me.
So go ahead, Manal. I was just going to pose one question to you because, certainly, Roger's raised the point that the payers would likely interpret the label if the label is, in fact, ends up being, biopsy-proven NASH of a certain severity. They would interpret that to their advantage, meaning they would restrict access as much as possible, unless there is pushback from the medical community. So I'm curious to know, and I'm assuming you were going to speak to this, but what sort of pressure or pushback or advocacy do you expect if drugs are approved for NASH in this way? Do you expect clinicians to be arguing?
So I think we can lean on history, and if we take a look at the hepatitis C story that so beautifully unfolded, is initially all the drug compounds moved from being highly ineffective to amazingly effective with nearly 100% cure rates. And although those trials using the direct-acting antiviral therapies or combination interferon ribavirin utilized liver biopsy to risk stratify patients in addition to genotype and hepatitis C viral load, ultimately, what happened is as the drugs became highly effective, 100% cure rate with shorter durations of therapy, 8 to 12 weeks, despite the labeling, it almost became a little ridiculous to consider a biopsy when you're going to have a highly effective way of curing somebody and preventing downstream complications of cirrhosis and transplant and liver cancer. So we completely fell away from the practice of even doing a biopsy in the hepatitis C patients, and that became a more easier argument as the cost of these therapies started coming down. I don't recall the last time in the past few years that I've done a liver biopsy on a patient with Hep C. I've just ordered antiviral therapy and treated effectively.
But you brought up oh, go ahead, sorry.
And so it really becomes the balance of the interventions we're doing balanced with the cost and safety and efficacy, really, of the compounds that we're using.
Okay, and you brought up a great point because you're comparing or you're discussing HCV and the advent of these drugs with high cure rates, but now we're talking about a situation where we have patients that are asymptomatic for the most part, patients with certainly early in, F2 fibrosis. I don't know about the F3s. Cirrhotics, of course, would be symptomatic, many of them. But you have many patients that are asymptomatic, and you have drugs that are providing a modest benefit over that, some of which may have, fairly significant side effects. Again, just, not to pick on Intercept, but, with OCA, there is the pruritus and the elevations in LDL. So, in that situation, does it become, is there less of an argument then, of course, it sounds like there is, to push for, less stringent requirements on the use of these drugs? So, Manal or Roger, either of you, if you could comment?
Okay. Sorry, I was waiting for Manal. Do you want to go ahead, Manal, and I'll follow on?
So is there a push for less stringent requirements? I am of the opinion that the push for less stringent requirements is going to be driven by the risk that the patient has. When you take a look at the natural history of this disease, it is a disease that spans, 20, 30, 40 years, depending on the rate of disease progression and the comorbidities that patients have. And so it doesn't necessarily make sense to treat a patient with NASH who maybe even has stage 0 or even stage 1 fibrosis if what you're treating for from a morbidity and mortality standpoint isn't going to catch up with them for decades to come. And effectively, if they were motivated enough, they could treat themselves with a good diet and exercise. And then to put them on compounds that may have associated cost, potential risks, or require concomitant therapies to treat side effects doesn't make sense. However, if you are to ratchet up the risk, and I am dealing with a patient who's got well-compensated cirrhosis, who, if I don't treat, may have hepatocellular carcinoma, which I have no therapies for effectively, or maybe just local palliation. And if a consequence happens to them and the time horizon for that consequence is shortened to on an order of 5 to 7 years, they might be looking at transplant. Well, you bet I'm going to be advocating for that patient, because the alternative to no therapy is death, transplant, or cancer, for which I have no good options. And then, irrespective of the market value of that compound, it becomes highly, highly cost-effective because the alternative is a $500,000 to $1 million therapeutic intervention for which may or may not happen for them.
Okay, and Roger, I'm gonna turn back to you in just 1 second, where we can talk about drugs that would intervene at different stages of the natural history of the disease. And Manal was, of course, talking about patients with more advanced fibrosis or cirrhosis. But before I do that, I just wanted to talk a little bit more about one point, and it's the point about how clinicians will identify patients in clinical practice. And I know, Manal, what you've said so far, it sounds like you're still talking about if the label requires, if the label is for patients with biopsy-proven NASH, that maybe that is the way that you would be forced to identify patients before treating.
No, I didn't say that. I didn't say that. That may be what I am forced to do as a provider to get a patient access to drug.
Okay.
I can identify patients in clinical practice and risk stratify based on age, over 45 or 50, the presence of 3 or more risk factors for metabolic syndrome. Certainly, if they're diabetic, in my own mind, even without that biopsy, they're more likely than not to have fibrosis on biopsy. And if their platelet count is below 150, then I'm pretty confident that they'll have advanced hepatic fibrosis. So the biopsy confirms my clinical acumen, or maybe complements other non-invasive markers like a FIB4 or an ELF or even a FibroScan. But if it's written into the label, and if third-party payers mandate what is in the label to pay out for these drugs, and that label says a liver biopsy, then that twists my arm as a provider to perform that liver biopsy to get access to drugs.
Understood, and thank you for clarifying, because that is something that I wanted to address, because many experts that we've spoken to indicate that they're using sort of the clinical picture, whether it's just the clinical attributes or putting them to an official algorithm, or things like FibroScan or other non-invasive imaging to diagnose and risk stratify their patients. And I understand the issue with the label, but what I'm curious about is what would we need to do? Would that be sufficient for you as a clinician to identify the patients? And it sounds like the answer is yes. But what would we need to do to prove to payers that that is sufficient? Because obviously, very few people are getting biopsied today. And I'm curious, Manal, if you can just speak to that. Is that is it 5% of the population of the patients that have NASH? Is it less? Is it more?
Well, yeah, I think you got to look at it. There's a bottleneck for access. With only 3,000 hepatologists maybe in the country, that are doing biopsies, given the population at risk that may need it, we just don't have, the resources by which to biopsy every patient who may be walking around with NASH and diabetes or 3 or more metabolic features of metabolic syndrome. So it's a feasibility issue, not to say that biopsy couldn't be done and done safely, but there is added cost, added risk, and access issues to such an intervention, which we haven't addressed on the opposite side, is access. If we have surrogate markers, certainly I could envision a algorithm or metric by which third-party payers would help us risk stratify, or not. If a drug is $80,000 coming in on the market, then that risk stratification would have to be pretty tight. But if the cost of the drug is comparable to an oral anti-diabetic medication or a statin, then who cares what the patient has, particularly if the patient otherwise, if they have risks for NASH or have evidence on biochemistry for NASH, and the profile for that compound is very safe. I don't envision that the landscape for treating NASH is going to be short and sweet. These are going to be long therapies with long-term endpoints for treatment.
All right, very good. And I did want to follow up just a little bit here. to understand then, let's say that, again, we're talking about a drug that might be around $15,000 just based on analyst forecasts or predictions of what the pricing is, sort of an average of what obeticholic acid or GFT505 might be priced at. Let's say something like that. And then if you were talking about a non-invasive methodology or some approach to risk stratify, what can you imagine might be the preferred approach in the future? And I'll ask Manal, you to speak to that. And then Roger, maybe you can talk about what would be required from the payer's perspective in order to support the implementation of that sort of replacement for biopsy.
I envision that the risk stratification is going to have to be a point of contact intervention. I do not envision that risk stratification is going to be ultimately by biopsy or by MR elastography or MRI, also because of cost constraints and access issues. I'm hopeful that we will have a serum biomarker or algorithm that can be coupled with an in-office FibroScan that can risk stratify patients into low risk, high risk, and if deemed necessary according to labeling, yes, a biopsy may selectively need to be done to get access to the therapeutics depending on the cost and the side effect profile for that. But I would envision a point of care contact. It needs to be done by a primary care physician, an endocrinologist, or a hepatologist ultimately. But right now, the seat is in the camp of hepatology.
Okay. And Roger, then I'll just turn it over to you, because if we're able to implement some sort of non-invasive diagnostic procedure for the most part, what would be required, do you think, from the payer's perspective in order to support that instead of going to biopsy then?
Well, first and most importantly, there would have to be some pretty significant medical consensus, probably written up in the guidelines from EASL or ASLD or something like that, that actually says, yes, these guidelines, these diagnostics, in fact, do risk stratify in a way that medical practice should follow.
Okay.
So that's my sense is that that's going to take some time. Indeed, I think that one of the key challenges for NASH is getting into or getting guidelines written, and with a host of new therapies coming out without a ton of real-world evidence, that is going to take some time. And by time, I mean years and years. Without those guidelines, it's going to be very difficult to pressure payers to move one way or another outside of the label.
Okay. And Manal, I'm curious if you have any thoughts on, we have ongoing phase 3 studies for both obeticholic acid and GFT505. Certainly a lot of secondary endpoints being included in those studies to support the biopsy results, resolution of NASH, improvement of fibrosis. I'm just curious, those are pretty long studies. They're looking at, after conditional approval, they're looking at long-term outcomes. Do you think that we'll have support from those studies, sufficient support from those studies to, justify any of the non-invasive techniques that we might use to risk stratify? I'm just curious if that data might come out of those studies.
I envision it will. I think those studies were well designed. They made thoughtful for consideration of endpoints, both clinically and trying to establish predictors of responsiveness to these compounds and surrogates to biopsy. Ultimately, with cardiovascular disease being the leading cause of morbidity and mortality with fatty liver disease. We don't want to treat patients to increase cardiovascular risk at the expense of treating liver disease. And at the same time, I think the FDA is justified, certainly, to better understanding what the intervention does for all-cause morbidity and mortality. We learned that statins decreased cardiovascular disease despite causing some muscle pain in a minority of patients. But we would need to have an understanding of what it does to the overall landscape of health.
Okay. So some of that may come out of those studies, but maybe some additional information required still to establish these non-invasive approaches. All right. Okay, so I think we've got a consensus here that at least initially the bar will be pretty high to support reimbursement and by virtue of that utilization in the clinic for many patients, and it may be restricted to the patients with as the studies have been designed with F2 and F3 fibrosis. I think it's still open about those F1 fibrosis patients, as there is an exploratory cohort there. But you may need to identify them based on biopsy. The payers may be able to, hold that position based on the way those studies are conducted. But over time, we may be able to loosen up those guidelines as we have requirements, as we have greater data that shows a correlation between the non-invasive methodologies, FibroScan, MRI, whatever, or other clinical parameters, as Manal, you pointed out, and outcomes. But some of that data may come out of the ongoing studies for obeticholic acid and GFT505, but it may take some while to implement. So that's very helpful. I think what I wanted to do now is move on to another question. We've started to address this. The question is, how will the pricing of drugs for NASH influence market access and reimbursement? And clearly, we understand that, obviously you raise the price, there's going to be, the bar is going to be higher for reimbursement for these drugs. But Manal, you raised the point of certain subpopulations or places within the natural history that could support a higher price. So patients with decompensated cirrhosis, for example, NASH patients with decompensated cirrhosis. So I just wanted to talk about this a little bit because clearly this disease is a spectrum. And, when we talk about, all the drugs in development, we talk about drugs for NASH, but really, depending upon the mechanism, depending on many other things, one drug might be appropriate for one place in the disease versus another, and accordingly may have, support different levels of pricing. So I just wanted to, see, Manal, if you can speak to this. You already did to the decompensated cirrhosis part, but are there other subpopulations of patients that might support a higher price? Patients that are, might progress more rapidly than others. And what about the early disease? There certainly are people, developers, looking into drugs that would inhibit steatosis. Do these have any place, by themselves? So I'll just let you answer that question, and then Roger, maybe you can speak to the payer perspective on that.
Well, I think we have enough evidence to suggest that isolated steatosis in the liver has no associated increased risk on overall liver-related morbidity and mortality. So when you ask a hepatologist, do you need to treat steatosis? The answer is no. It's not gonna impact one's liver-related outcomes. You ask a cardiologist if you need to treat cirrhosis, they say, yeah, sure, because the patient who has steatosis is at increased cardiovascular risk. Or an endocrinologist may say the patient who has isolated hepatic steatosis is at increased risk for new onset diabetes. But we're talking about liver-related outcomes with these registry studies and what the FDA is looking at. So from a liver standpoint, does isolated steatosis need to be treated? No. It's at very, very low risk for disease progression. So that puts us into the camp of NASH and NASH across the spectrum of fibrosis. Right. So we all agree that the more advanced you are, the more disease activity you have, your rate of disease progression may be higher or faster. So those with definite NASH or a NAS score of 6 to 8, with at least, stage 2 fibrosis is a patient who would be deemed high risk, even though they're not cirrhotic. Does that make sense? So, so we would be risk stratifying according to the phenotype of that patient. We can debate whether that phenotype is clinical or histologic or a combination of both, but ultimately, stage 3 fibrosis is a very high-risk cohort histologically, and if you take a patient with stage 2 hepatic fibrosis and add to the mix their hypertensive, dyslipidemic, diabetic patient who's 45, then one would be confident to say that's also a high-risk patient, even though they have a lower fibrosis stage. And so I think, yes, we will use the clinical and histologic phenotype to drive priority and access to treatment, and expand that priority and access based on concomitant comorbidities that may be helped by maybe some of these therapies. Certainly if a drug may improve glycemic control or improve lipids, maybe that they don't have to have as high risk from a liver standpoint, but they may benefit from a metabolic standpoint. If we know a patient drug is highly safe and cost effective, then maybe the funnel of access and priority to treat may drop from a stage 4 or 3 down to a stage 2 with moderate metabolic risk, or maybe even a stage 1, depending on the cost and safety profile of a drug. But from when you ask a hepatologist, fibrosis begets more fibrosis, especially when you cross the threshold of stage 2, you're unlikely to reverse spontaneously.
Okay, very good. Thanks, Manal. And Roger, I'm just curious if you have on that. How would the pricing of drugs influence market access and reimbursement? Is there room for premium pricing or higher pricing, at least in patients with more significant disease? If a drug is specifically developed for patients with more advanced fibrosis, for example.
Right. I mean, I agree with what Manal is saying. The higher the risk the patient, the higher the provable risk for the patient, probably the higher price that can be justified, with the following caveat. Payers are going to be very concerned about leakage. That is to say, moving from the very high, the provably high risk patients into other populations. So granted that Manal and other hepatologists could say, well, all right, this drug improves glycemic control, or it improves other cardiovascular issues. We're going to kind of relax our prescribing and move to patients with nominally lower risk, lower fibrosis scores, for example. Yeah. Payers are going to look at that and say, wait a minute, before you do that, before we allow that kind of prescribing, we're going to demand from the manufacturers outcomes trials which demonstrate that those drugs are in fact clearly valuable in those cases. And those outcomes trials are not going to be cheap.
Understood. And I'd like to move on to the next question, because we've been talking about pricing and the impact on access. But one thing that we've heard time and time again from the experts that we've interviewed is the potential for combination therapy, that a single agent may not be sufficient for all patients, and we may have to add treatments together with different mechanisms of action. So I'm just curious then, what are your thoughts about once multiple drugs are approved, presuming clinicians would recommend combination therapy, are payers going to manage access to these combinations?
Right. I think it's an interesting question, and I think that it's a relatively rare situation where you see a single disease managed by multiple drugs, multiple branded drugs. I'm sorry, I should let me say that. Clearly, diabetes is managed by multiple drugs, but most of them are generic. Once you have multiple branded drugs, as in, for example, oncology, payers start to look for alternatives. The reason they can't, oncology, they simply can't. It's a protected class, and the government simply doesn't allow restrictions in oncology in the same way they do in other areas. Here, however, they will definitely be very careful about this, and my bet is that what's going to happen is they are going to start looking for particular drugs in common therapy. Say if there are multiple FXR candidates drugs out there, they are probably going to try to focus on a single one, drive the price of that, the net price of that down, and require the use of that drug in all combination therapies that use an FXR candidate drug. So one of the things that's going to be important for developers is to understand where their drug is going to fit within a combination and what competition they'll have, because my bet is that there will be exclusive contracts within those categories to make so that the payers will be able to at least control price that way.
Okay, thank you, Roger. And that does depend then on multiple, in-class competitors then being a backbone of therapy. So I want to turn this over to Manal for a second. What are your thoughts on combination therapy for one? That being the way of treating patients in the future, and is there any sort of leading candidate for a backbone yet? I know there's a lot of obviously a lot of activity in the FXR space, certainly in the anti-diabetics, but I don't think there's a single class that's where that's favored, and there are multiple competitors. Any thoughts about that moving forward, where there may be a situation where we have a favored backbone regimen, that there might be multiple in-class competitors?
Yeah, I mean, from a liver standpoint, this is a very heterogeneous disease. We got different histologic features to it, and certainly one size doesn't fit all. And one could argue that while you may be able to treat fibrosis, unless you're treating the underlying root cause for why that fibrosis is there or has gotten there, then you may not actually be doing your patient a service. You're leaving a runaway freight train and just trying to control the aftermath of the injury, which is fibrosis. With hepatitis C, it was different. We had liver injury that led to necroinflammation of the liver and fibrosis. There was a unique target, and that unique target is the virus. And in NASH, the unique target is altered metabolism. Or it could be lipids in some people, diabetes in others, maybe weight in others. So the clinical phenotypes are very heterogeneous. So I could envision a paradigm where we're treating patients with an antifibrotic therapy and then a tailored approach to their modifiable risk. And so I don't think we're going to get away from a multi-drug therapy. I could envision that the backbone of treatment could be a drug that could be effectively used long-term with minimal side effects. Potential therapies that could be long-term use, maybe PPAR compound, could be an anti-diabetic, anti-lipidemic therapy that could be used long-term and safely. could be a compound like Centercivaroc. So I think time will tell, but certainly if we move to long-term therapies, the backbone will need to be cost-effective with minimal side effects and minimal need for routine surveillance monitoring.
Okay, very good then. So certainly moving towards multi-drug therapy, but it may be a different backbone for different groups depending upon the underlying metabolic defect because of the heterogeneity of the disease. And then the therapy, of course, the backbone would need to be very safe and well tolerated. So that's very helpful. So I'm gonna move on to the very last question now. We'll just pose this question to Manal and Roger, and then if we have any questions from the audience, we can certainly take that. But so the last question really I had is to, given the sort of the obstacles that we've discussed, there's certainly going to have an impact on the size of the patient population that can be treated. And we're going to have an increasingly competitive environment. We've got, as I said, now it's over 250 drugs in development, a lot of activity here. We're definitely going to, well, hopefully going to have some drugs on the market in the fairly near term. So what can companies who are developing drugs for NASH do now to sort of help realize the full potential of this market in order to, stimulate uptake and potentially overcome some of these reimbursement hurdles? And I'm just curious, maybe Manal, we can start with you, and then Roger, we can turn to you to talk about the payer's perspective.
Well, I think realizing the full potential of the market is a challenge because what we're hindered with is education and understanding. It still amazes me that many healthcare providers have not realized the progressive nature in unique cohorts of this disease and may consider NASH still a benign entity, right? And so while the best understanding sits in gastroenterology and hepatology, we still haven't moved forward where amongst patients with diabetes, liver enzymes are routinely being performed to even screen for NASH and NAFLD, except for those that are maybe at, larger centers have heard about this. But I certainly don't think it's being done in obesity clinics where every time you come in, you're overweight, you get a set of liver enzymes drawn. So I think education and appropriate guidances and guidelines on who to screen, we have to overcome that hurdle before I can even answer this question. But I think education, that societies that are invested in the space of obesity, metabolic syndrome, and liver disease, write unified guidances and guidelines by which to lend practitioners insight and better understanding.
Okay, very good then. And Roger, maybe you can speak then again to the from the payer's perspective, what a company might do to prepare for entering a very competitive market to facilitate reimbursement and better management of their product. What can companies do today?
I think that there are probably three major things that companies have to think about. The first is that they've got to focus on an easily, readily identified target population, a target population which a payer can define the prevalence of early, and that allows the payer to create rules around. So if there's an easily understood protocol for that particular patient, great. It also has to be able to, that system has to be able to minimize what I'd call leakage, using the same drug in other populations. So how can you deal with that payer issue? The second issue is that I think companies need to start thinking today, now, yesterday, about some kind of value-based contracting. And that means figuring out what are the endpoints that can be tracked that demonstrate whether the drug is working or not working. Payers are going to be increasingly interested in that, and that has to be done very practically. Finally, and the third thing is that I think that drug companies have to recognize that payer economics count more than the health system economics. And it's not that health system economics don't count, but it is to say that payers are private, generally private organizations with their own P&Ls and own budgets, and they're not going to be seeing the world in the same way that the overall health system would. Indeed, there is no overall health system point of view because there's nobody controlling the overall health system. So, for example, no payer is going to be particularly interested in an argument that 7 years from today, well, actually, a few payers may be, that 8 years from today, we're going to be saving somebody from a liver transplant because they're not going to be paying for it. Unless those patients are in Medicare Advantage plans where patients stay, from 5 to 7 years within the plan, most plans have a beneficiary life of 1 to 2, maybe a little bit better than that, years. So the payer economics are a much shorter term issue. So making the overall health system economic argument doesn't really help today.
Understood. Roger, thank you so much. And, I'd love to continue the conversation, but unfortunately, we're running out of time here. So we did have a few questions that were submitted online, but I think we're going to have to address them directly after the webinar. I just wanted to close by, first of all, thanking both you, Roger, and Manal for participating today. It was really informative for me, very helpful. But any final thoughts from either of you?
You've gotten my thoughts.
Thanks, Roger. Manal, any final thoughts, or did we capture pretty much everything through the conversation?
No, I think we discussed a lot of hot issues and captured quite a bit this discussion.
Okay, great. Well, again, thank you both very much, and thanks to everybody for joining today. We will follow up with any questions we receive after the webinar. So thanks again, and enjoy the rest of your day.
So long, everyone.
All right. Bye-bye. All right.